The central dogma of molecular biology, from DNA to proteins. In this video, we will explore the central dogma by looking closely at how all things start from DNA, are copied into RNA, and produce a protein. This protein then in turn becomes some sort of expression tool, showing a trait, performing a function, or something else. But the bottom line is that the processes of life depend on these proteins being made. This fundamental principle explains how genetic information flows within a cell to create the proteins that keep us alive. This process occurs in all living organisms, ensuring that life functions as we know it. DNA, the blueprint of life. DNA, or deoxyribonucleic acid, is the molecule that stores genetic instructions. It is often referred to as the blueprint of life because it contains the codes that dictate an organism's traits and cellular functions. DNA is composed of two complementary strands that twist into a double helix structure. Each strand is made up of smaller units called nucleotides, which consist of... There are 4 nitrogenous bases in DNA. Adenine, which binds to thymine, and cytosine, which binds with guanine. This strict base pairing ensures that DNA can be accurately copied and transferred from one generation to the next. The sequence of these bases forms genes, which carry the instructions for making proteins. DNA replication, copying the code. Before a cell divides, it must make an exact copy of its DNA so that each new cell gets the same genetic information. This process is called DNA replication and occurs during the S phase of the cell cycle. How DNA replication works. Unzipping the DNA. An enzyme called helicase breaks the hydrogen bonds between base pairs, separating the two strands and forming a replication fork. Building new strands. Another enzyme, DNA polymerase, reads each exposed strand and adds complementary nucleotides to create 2 identical DNA molecules. Proofreading and correction. DNA polymerase also proofreads the new strands, correcting any errors to prevent mutations. Since each new DNA molecule consists of one original strand and one newly synthesized strand, DNA replication is known as semi-conservative replication, half old, half new. Transcription. From DNA to RNA. Now that DNA is replicated, how does the cell use this information? The first step in turning genetic instructions into proteins is transcription. What happens during transcription? Initiation. Inside the nucleus, an enzyme called RNA polymerase binds to a specific gene on the DNA and unwinds a small section of the double helix. Elongation. RNA polymerase reads the DNA sequence and creates a single-stranded molecule called messenger RNA, mRNA. Termination. Once the mRNA strand is complete, it detaches from the DNA and exits the nucleus through a nuclear pore, heading toward a ribosome in the cytoplasm. Unlike DNA, RNA has some key differences. RNA is single-stranded, while DNA is double-stranded. RNA contains uracil U instead of thymine T. The sugar in RNA is ribose, not deoxyribose. mRNA serves as the messenger, carrying genetic instructions from DNA to the ribosome, where proteins are made. Translation. From RNA to protein. At the ribosome, translation begins, converting the genetic code in mRNA into a functional protein. Steps of translation. Initiation. The ribosome attaches to the mRNA and starts reading its sequence in groups of 3 bases, called codons. Each codon codes for a specific amino acid. Elongation. Transfer RNA, tRNA. Molecules bring amino acids to the ribosome, matching their anticodon with the correct codon on the mRNA. Peptide bond formation. As amino acids are delivered, they link together, forming a polypeptide chain. Termination. When the ribosome reaches a stop codon, the process ends, and the completed polypeptide folds into its final functional protein. Proteins are responsible for nearly every function in the body, from building tissues and enzymes to regulating chemical reactions and immune responses. The significance of the central dogma. This entire process, DNA to RNA to protein, is known as the central dogma of molecular biology, a concept first described by Francis Crick in 1958. It is a universal principle that applies to all forms of life, from bacteria to humans. This flow of genetic information ensures that cells grow and function properly, repair themselves when damaged, and pass genetic traits to offspring. Without the central dogma, life as we know it would not be possible. Exceptions to the central dogma. While the central dogma is a guiding principle, there are a few exceptions. Reverse transcription. Some viruses, like retroviruses, e.g. HIV, use an enzyme called reverse transcriptase to convert RNA back into DNA, defying the typical flow of genetic information. RNA editing. Some organisms can modify their mRNA sequences after transcription, leading to different proteins from the same gene. Prions. These are misfolded proteins that can transmit information without DNA or RNA, causing diseases like mad cow disease. These exceptions expand our understanding of genetics and molecular biology, leading to breakthroughs in medicine and biotechnology. Conclusion: life's molecular symphony. From storing genetic blueprints to producing the proteins that sustain life, the central dogma is the foundation of modern biology. Understanding these processes helps scientists develop new medicines and gene therapies, study genetic disorders and their treatments, explore synthetic biology and genetic engineering. Whether in medicine, agriculture, or bioengineering, the central dogma continues to shape scientific advancements. Thanks for watching, and remember, your DNA is the code that makes you uniquely you.